Hi friends, today we can discuss how we can study or how people have studied membranes. We are looking more into membrane biology today. Electron microscopy has provided the biologist a detailed portrait of the structure of the cells. Usually electron microscopy in earlier days have provided the ideas about the structure of the cells, but they have not given much insight into the function of these components. So by combining both structure and function of the membrane structures only, we can conclude how they work, how they transport materials, how they function, what are their components, what are whether the membranes differentiate, whether membrane of one organelle differentiate with other, like that. Only structural component can provided by the electron microscopic images. So we need more better techniques in order to look into the membranes. Autoradiography is the first and most earliest technique used in order to elucidate the function of the endomembrane system. Pallet and Jameson once incubated the pancreatic tissue in radioactive amino acid, so that the protein synthesized in the tissue will be radioactive. Then they transfer it to normal amino acid media. This experiment is known as pulse chase. As in pulse, the radioactive amino acid get incorporated in the protein. The protein synthesized using chase are contain non-radioactive amino acids. So, as the longer the chase, the farther the radioactive proteins manufactured during pulse will travel from the site of synthesis within the cell. We can fix the cell at different times, and we can look where the location of the cell, where the protein is synthesized, where now it is transported, like that. Location of the cell, particular protein or cargo can be determined by this experiment. We can see it in this slide in the figure A, which represent the autoradiograph, and figure B we can see 3 minutes it is just synthesized from the rough endoplasmic reticulum, and 20 minutes it is transferred to the Golgi complex, and about 120 minutes it is transported or secreted out. So we can find out the mechanism of membrane transport by this autoradiography, by this like this. Another technology is the green fluorescence proteins. The fluorescent protein which is produced by jellyfish, which is further utilized in tracking the proteins or cargo inside the cell. Here the DNA encoding the GFP need to be fused with the DNA encoding the protein, which need to be studied, so that we can see in the live cell we can see the migration of this particular protein from the cytosynthesis to where it need to be destined. Once inside the cell, the chimeric DNA expresses a chimeric protein which consists of GFP fused to the end of the protein to be studied. That is, the protein which produced there will fluorescent. As the protein which target which get expressed, it get fluorescent green because it is a chimeric protein now fused with the GFP. Here what we see green in this figure is the endoplasmic reticulum. The protein which forms in the endoplasmic reticulum get concentrated in the endoplasmic reticulum. So the endoplasmic reticulum appears green because the chimeric protein which is fused with the fusion product of both GFP as well as the product which is targeted is now expressed abundantly in the endoplasmic reticulum. So the endoplasmic reticulum appears green in color. Autoradiography as well as electron microscopy as well as GFP techniques can provide only more information about the structure and function of the cellular organ, but they fail to provide information about the molecular composition of the structures. Actually, biochemical techniques were utilized for finding the molecular composition of the structures, how the membranes differ, whether the endoplasmic reticulum membrane is same with the plasma membrane, or whether the endoplasmic reticulum membrane is same with the Golgi complex, whether the rough endoplasmic reticulum membrane and smooth endoplasmic reticulum membrane is same. For that, mainly density gradient centrifugation is widely used. First, in first centrifugation, the cells are homogenized and centrifuged so that the bigger organelles such as nucleus, mitochondria get separated by the first slow speed centrifugation, that is about 20,000 G for 20 minutes. Then we require a 50,000 G for 2 hours, a longer duration, a higher RPM or higher G required for the next centrifugation so that we can separate the rest of the membranes. Usually when breakout, when the cells get homogenized and membranes get break, they form 100 nanometer size vesicle known as microsomes. These microsomes may differ each other between plasma membrane as well as endoplasmic reticulum types, so Golgi complex. The microsomes of the rough endoplasmic reticulum is surely different from that smooth endoplasmic reticulum, as rough endoplasmic reticulum microsomes contain the ribosomes. So by this biochemical technique only, by this density gradient technique only, it was found for the first time that the protein synthesis, primary site of protein synthesis is rough endoplasmic reticulum, and something which make the microsomes, that the endoplasmic reticulum microsomes were rough, which is the main thing involved in the protein synthesis, that is the ribosome. Not the membrane. Membrane has no role, but the something which make the membrane rough, that is the ribosome, which is involved in the protein synthesis, was found out for the first time by this density gradient centrifugation. Using mutated proteins, that is secretory proteins, which are involved in the secretion can be mutated. This figure in the slide is an yeast cell. These are 3 yeast cells. The first cell is a normal cell, and second and third is a mutated cell. So first cell, it looks normal, but second and third, which produce the proteins, first cell we can see some vesicles which transport the cargo, but second and third cells there is no vesicle, there is no transport. They are mutated to particular protein, where is 17, which is involved in the membrane transport. So their morphology itself reflects the defect in transport mechanisms. So we can likewise we can understand which proteins are involved in this transport like that. So these are some of the techniques involved in exploring the membrane structure, function, their molecular characteristics, everything. So we will come with more videos like this. Please subscribe to this channel. Thank you for watching. Thank you.